Hello and welcome everyone in another episode of Researcher Celebrity. Today we have Dr. Anandi Karumbati with us as our researcher celebrity. A brief introduction about Dr. Anandi. She has done her bachelor's in pharmacy from BITS Pilani. For PhD, she moved to United States and attended at University of Kentucky, where she also did her postdoctoral fellowship research at University of Michigan, and then moved back to India and joined RIA Metrics as senior scientist and then also served as project lead. Then she moved to C Camp and joined as lead scientist and also worked as senior translation manager. And currently she is team lead for molecular and cellular biology at CCBT InSTEM Bangalore. With this brief introduction, we'll say thank you so much, Dr. Anandi, for taking time out.
Yeah, thank you so much for having me here. And I've seen your episodes and it's wonderful. Yeah.
Thank you so much for your kind words. We
Yeah.
always start these conversations that how and when you decided that you want to become a researcher.
Um, I was okay. It's a long story. So actually, I just went back to my old days and I was looking at my yearbooks from high school and Bits Pilani. And it's amazing because there was one subject that caught my eye, and I always wanted to be in that stream. Like, I wanted to be in genetic engineering. I don't know, somehow that word was probably one of the buzzwords when I was studying or something. I didn't know I was going to get into research. Honestly speaking, I thought more when I started pharmacy, like everyone else, I thought I was going to go into the pharma world, go into industry and move in that path. But life had other plans for me. So that's how I would say I'm here today.
Absolutely. So when you mentioned about the genetic engineering part, that was the infancy of genetic engineering when you were
Pretty
fascinated
much.
with this world. So it somehow signed up for you being a researcher because
Yeah.
at that point of time it was like the entire field was just starting.
Very nascent.
Where
I still remember some of my thing with agrobacterium and all in my BITS Pilani days. And incidentally, though I took pharmacy, a lot of my electives, I actually took genetic engineering. So that's the advantage of BITS Pilani. So you can have your, tailor make your journey. So I took genetic engineering, I took recombinant DNA technology. My projects were all in, mostly in the biology side more than
Mhm.
pharmacy. Though I said no, though I'm back to full circle, I'm still in pharma today. So
Absolutely. This is like, once you commit to a research question, it might just take tangents, but the core remains the same as you mentioned.
yeah.
So let's start with the story of when you were done with your BITS Pilani graduation,
Mhm.
bachelor's. The first thought that you want to do a PhD in United States.
Actually, no. I had so the bits also that had a good thing because we have something called a practice school, where we get to do internships in different places. So two of my internships, one of them was in a place called Cancer Research Institute in Chennai, and there I met a professor, Professor Ramesh Gupta from the University of Kentucky. And I saw what he was doing. He was doing something called P32 post labeling to see DNA adducts. Though we didn't do the radioactive part of it, we actually did the the cold lab thing, and that kind of was very fascinating. And at that point of time, I actually, it's not email or anything. Remember those days? So I actually wrote a letter. I didn't even have his address or anything. As I was finishing the graduation, I did in those days what was very, very actually new again. I took a gap year, which nobody would even imagine in those days. And while I was in my gap year, because I was all 4 years, I was in Pilani and I wanted to be in Chennai for some time before, which is my hometown. I wanted to be there with my people for some time before I embarked into a journey into the United States. And in the meanwhile, I did try some to get into the PhD programs in India. Did look at a thing, but still back of my head, my meeting with Ramesh Gupta and the kind of exciting science still stood in the back of my head. I actually wrote an email to him. I mean, not an email to him, a snail mail to him. And believe it or not, after 2 months, I got a response. And he was like, I wasn't even sure I could apply to this program because it was University of Kentucky Graduate Center for Toxicology, affiliated to the School of Medicine. So I wrote him. So after that, he said, yeah, yeah, you can go ahead and apply. And that's what made me, think about the whole process. So I wrote GRE, the subject GREs, and then wrote all the went to the the consulate, picked up the address and did all of those work and actually applied the long way, which now takes probably minutes to apply. So yeah.
Yeah, those were fascinating days, when you
And
don't
actually
have
went back to Pilani to collect my reference letters, by the way, to tell my professors.
exactly. Now it is like you write an email before you sleep, and by the time you're awake, you have the response most of the
Yeah.
times. But that's exactly what you are saying. This the our current audience, probably some of them can correlate with what we are talking about. Mostly
Mm.
they are like, why it was that slow process? But yes, that the technology has transformed our field like
Yes.
nothing else in this world.
That
So,
was the beginning of email, actually. Just then
mhm.
emails were starting.
Yeah,
So
yeah, yeah. So when you accepted the PhD at University of Kentucky, the first challenge you faced when you were there, and then how the journey went.
I'll tell you, even before I joined, my first condition was I should get full scholarship and admission fee waived before I go because I was going from India. So I was
Mm-hmm.
like, I have to have a full scholarship, and Kentucky was really good because I mean they gave a nice admissions plus financial assistance as well as research assistantship. So what you go there and do is, I embarked and went there, and I saw some alumnus of my BITS Pilani there as well. So BITS has a huge alumnus network. So it was nice. I reached out to my seniors and went there, and biggest challenge was, we were only seven people who were admitted into that program. I was probably the only Indian in that batch and only Asian in that batch, only women in that batch. So it was a different world for me. In that particular batch, we were seven of us, and I was the only, yeah, both different. So it
Mm-hmm.
took, and there were people coming in from engineering backgrounds,
Mm-hmm.
variety of backgrounds, and I didn't even realize that somebody from engineering could do a PhD in toxicology, which is what I did. And
Absolutely.
yeah, it was nice, and it was a different world. So getting adjusted to the way of working. Uh, Very funny episode was one morning. My professor, we had to do something called rotations. We had to
Mm-hmm.
do actually rotation. The first year was devoted to entire rotations. So we went and did four. I did 4 to 5 rotation. One of them being Professor Gupta's lab as well, whom I met in Cancer Institute. It was actually a very nice thing there, in the sense that though he was one of the connections by which I went to the program, he didn't necessarily stay, you have to stay in my lab. He was like, go choose what you feel really like doing, what you feel like doing, and then join that lab. I think that was really gracious of him. And even today, he has been a part of my thesis committee, and even today I'm in touch with him. And I think that was a very, very nice thing. Not many people do that. And
Absolutely. And I think now this is the time when we do talk about networking more importantly than any, like ever in research. We all have a small community, and no
yeah,
matter which part of the world you are doing research, you always
yeah,
know either people by their face or mostly by their names.
yeah.
Yeah. So when you mentioned about this first meeting with Dr. Gupta, you were sure
Mm.
that you want to join him as a PhD mentor, or it was just like, okay, you were fascinated by the work?
I still did what, by the way, I was just in a neighboring lab, and part
Mm-hmm.
of my work did involve adults, and he was very much a part of my thesis committee. But what I went there and saw is he was mainly focusing on DNA addicts, and that's it. So I wanted to do the entire. That got me interested in what I did there eventually, because I wanted to do genotoxicity, and that's what I ended up doing. So I set up a whole cell culture lab in a lab that was classic. So I ended up joining after the years of 1 year of rotation across various labs. I did across 4 labs. and eventually settled down in Dr. Larry Robertson's lab, which was exactly adjacent to Dr. Professor Gupta's lab. So, and a part of my thesis was in Professor Gupta's lab. Like I did do a little bit of ADX there, but I
Right.
expanded it a little bit, did more of cell-based assays, and that's where I started cell culture. So, and
Nice.
this was a lab that was heavily, heavily into in vivo work. Toxicology, it was always, the in vivo modules, and
Mhm.
it was very set. And we were looking at environmental carcinogens, like
Mhm.
polychlorinated biphenyls, and seeing the effect on DNA. I mean, seeing the effect on liver toxicity. That was the entire lab theme about. I was like, let me go ahead and look into the molecular mechanisms. I was very interested in dissecting how they were toxic, not just to know that they were toxic, but how they were toxic. The entire
Mhm.
initiation process, the mutations, I was more interested in that. And that's my entire PhD work actually. So,
So I would like to say this to audience that whenever you are going to research meetings, either it is conference, symposium, and you have this chance to interact with the supervisors, probably can be your future supervisor, always keep an open mind that, as Dr. Anandi mentioned, even if you are talking to someone doesn't mean that you are going to join their lab, but what actually fascinates you, you have to look for your calling, that you should not commit to someone just because of their name or just because of their uh, you know l- publication waste. There is so much which should go to the thought before you commit to a PhD, because it's a long long time.
should be
And
absolutely. I couldn't agree with you more. You should absolutely be passionate. You should be able to wake up in the morning and say, "Yeah, this is what I want to do today." So yeah.
absolutely. So let's talk about the uh, most common thing which happens in 99.99% of the PhDs, the failed experiments, the
Oh.
stresses brings, and how you coped up with it.
Um, I so I'll tell you. So we had to do. We were working on environmental pollutants, right? So it's a part of the EPA and all that stuff. So whatever we did would go into an MSDS. That was kind of lucked out a little bit there. So if we found that this kind of we found that okay, I did this, and I tested it in these experimental setups. For example, I would do micronucleus assay, and I would do comet assays. So all these assays I would do to test different metrics of genotoxicity. And then I would find that, okay, whatever I say what it is. And that's something which everybody should do. You shouldn't say a result just because your mentor wants you to say the result. You say it as it is, and probably it will lead to a different direction in the whole research. That's something I would like to tell the audience. So we just said what it is, because if it is toxic, it's toxic. It's non-toxic, it's non-toxic. Because you believe it to be toxic, or because a huge grant is written up on toxicity, doesn't mean you have to claim that. You say as it is. As long as your experimental controls are fine, and as long as you're doing it, you're technically you're fine. You just report what it is, and you can talk to anybody in my lab. Even today, they say I would be probably saying this 10,000 times. As long as your experimental setups are is fine, and within the scope of that experiment, you just report what it is. I mean, that
Absolutely.
way
I
you
think
knew.
No, I think that's where people do talk about, like, do not marry a hypothesis. Okay, you should be approaching a hypothesis with open mind. It
Yeah.
might follow as you think, or it can just go opposite of that way. And I think that is where most of the interesting research actually comes out, because
Yep.
it is not following the path which we believe should follow.
Yep.
Okay, so then after the PhD and postdoc United States, what made you move to back to India first?
I would say the reason everybody does family.
Okay.
Family, and I
So,
also had a small daughter at that point of
mhm,
time. So,
mhm,
and that's what actually that's my marriage is what led me to Michigan. So,
mhm,
my husband was working in the automotive industry, and I was working at Ann Arbor. My journey continued. I didn't stop. For example, I was looking at causes of mutations in my PhD, And throughout my postdoc, I was looking at the repair of these mutations, the innate repair processes in the body. So
Mhm.
it worked in the oncology space. But when we moved back to India, I was like pretty clear that and it was a very hot topic in the US, by the way. Everybody was they wanted to get the next scoop. Like, for example, they wanted to publish the next DNA repair enzymes. That was again the start of a very, very hot topic. So everybody wanted to say, okay, I found this enzyme, and they would give these cool names to these enzymes like Artemis, Apollo, and I was working in one of those labs, by the way. So they would give all these nice, nice names. And that's the time I really moved back, and I wanted to be in something that was more that switch. Actually, it was a lot of switch. One is a country switch, and second is also the career switch. I moved from academics to industry, and I wanted to be in a field that was relevant to India. So at that point, I don't know, that's when the infectious disease thing came upon me. So, and yeah, so that was a huge switch again. You can imagine going from a classic finding the next enzyme kind of labs to, going to DNA DNA repair to infection. That was a huge switch actually. So,
Absolutely.
yeah,
So when with this intent of giving back to community, you moved back to India.
yeah.
Now, let's take a minute to talk about when people do talk about in India now, and it is getting bigger and bigger, women in STEM. Your experience in United States and in India, how they were different, and how you see this changing from your BITS Plani days to current days.
I think a lot of people are interested in STEM, uh, but I feel, especially women, it's kind of the thing is changing. I told you, right? I was one of the one women in my program and the one girl. But soon after I joined, there were a lot of women slowly joining PhDs in toxicology. And back here, I came to see this. One thing I would like people to think about is, at some point when you feel that research is your calling, especially women, I think they should finish their PhD. I
Mhm.
feel a lot of youngsters stopping at masters and, trying to get their footing in masters. I think if, and biology, and whether you go into biomedical sciences or whether you go to biomedical engineering, if you want to stay in research, I'm not saying talking about the industry. If you want to stay in research, I think it's better you finish your PhD. That would be my thing, take it all the way through. That is something which I would like to say, that I think slowly people are also realizing, I hope so, and getting there. So it's a long journey, but it's worth it. It's not for the degree or anything. Even if you are masters, you're going to do the same work, might as well get the degree for it. So,
Absolutely. No, and I think the support Indian government is providing, especially to the women candidates, it also boosts them that, okay, they have the passion to pursue it. Now they have the financial means to support the family in case if they have to. So they should do that. But when we
yeah.
do talk about this, like the challenges, people always talk that, okay, when it is about starting the family, women
Mm.
have like these are one of those things which is still a taboo, and people think about it. Although the things are changing in India, the situation, circumstances, now we do have more PhD scholars. Like in some of the institutes, actually, girl number outnumbers the males, which is a good change. But again, with this, as you mentioned, that some of them either do not complete their PhD or drop out, or after their PhD, they just marry and do not pursue the research. For this, what are your suggestions or guidance to them?
You should join some institute and contribute the way you can. I think it's, if you call going to the United States or some other country a brain drain, this is also a brain drain
Mhm.
because you have
Absolutely.
invested a lot of your time, everybody's time into your career. I think whatever be it, whether it's engineering or biology or whatever you take, I think you should give back in some sense what you're trained for. In some way, you should contribute. I mean, definitely women. I think so. That's my personal opinion. But I'm also lucky. I have had like very good supporting. My house front has been extremely supporting of both of all my career choices. So, and I must say, I'm happy to have that. I wish more people, especially the current generation, support each other and bring each other up, So it shouldn't leave to permanent gaps, is what I
Yeah,
feel.
absolutely. And I think this is the time when we do talk about this, that either female or male, when it comes to research, research does not know any gender bias.
Yeah.
Whosoever is doing the experiment will follow the basics and the results will outcome obviously like that. So for all the male researchers also, like if your uh, better half happen to be a researcher too, nobody knows better than you that what are the challenges it brings and how you accomplish things together, how you can. But even if for the spouses who are not in research and happen to have researchers in their better halves, they should support them in a way. Either it is parent before the marriage or after the marriage. These, as Dr. Anandi mentioned, that every PhD takes so much of effort, not only by physical efforts or mental efforts, but also about the society have provided those means, either the institutes or the taxpayers' money, whatnot, all it comes together. So, with all the training, they should also give back to the community as they can the best way. As you mentioned, that either academic, industry, wherever you want to go, just go there and give it.
There are so many online resources also, you can do a tutoring if you're not able to get out. That's what I
Mhm.
feel like. There are so many opportunities now, not like
Yeah,
as I.
absolutely. The technology has transformed it so much. Like now there are freelancers, like researchers after their PhD, they're like, okay, I do not want to join anything, but I'll utilize the training which I had from my home via all these electronic technology. And I think that is also one of the way. Do
Yeah,
whatever makes you happy and make the most of it. So now
yeah.
moving back from academia to industry,
Mm.
then the perfect translational C camp, which is known worldwide, I'm sure. And then now as a team lead, this journey, if we can talk now.
Yeah, sure. So when I was finishing, so the industry, let me just talk a little bit about the industry. I was working in the industry, as I told, in the area of infectious diseases. We were making like diagnostic reagents for a instrument in making. So this was a new sort of an imaging microscope that we were building, and the company was based out of actually San Francisco, Dr. Balamanian, and he kind of designed this instrument, and I was in the biology side, so I was kind of making the reagents for that. So it was working with a cross-disciplinary team, with the digital image processing expert to an actual person who makes the hardware, and it was actually a very fun journey. And from there, after a point, the instrument keeps upgrading. You will you will get the newer versions, you'll get a beta, you'll get a alpha, beta, whatever versions of the instrument. But my growth will be like, after some point, it was okay, I'm done with it. Like, I want to do some more science. So that's around the time I met Dr. Taslim Arif Syed, who is now the CEO of CCAM. And he was like, why don't you come to here? So he had a project on early drug discovery, and it was on a new novel target in HIV. And this was a target which was found by all the stars in India. So it was by Professor Satyajit Ra, Dr. Satyajit Rath, Professor Satyajit Mayer, then Dr. Shai Jamil. So they all identified this novel target by which the virus evades the immune system. So he's like, since you have worked a lot on HIV, why don't you, and you have a pharmacy/toxicology background, can you come and help us with, validating this target? And actually, that started a decade ago. I still do the validation with me. Target validation is what I do even today. So when you get a pathway or something in any science, for example, it could be a cancer pathway, it could be a virology pathway, it doesn't matter. So when you find that a particular Achilles heel of the virus is there, or a particular protein is there, which you can actually come in with a chemical You won't call it a drug. You can come it with a chemical probe or a chemical thing at that point, intervention at that point. Then you're kind of disrupting that pathway and you're creating a drug. That's how it works. So if the properties are good enough, it becomes a drug. Otherwise, it can still be used to dissect the biology of the protein as well as slash cell process. So I was kind of excited because this coming back to drug discovery and my old origins. And I was like, okay, I'll take it up. And it was a very, very challenging thing actually. I call it my second PhD because this viral proteins are really, really nefarious. And the protein
Mhm.
was NEF, by the way, the protein I worked on was NEF, is NEF. And so we always call it nefarious because they are built to kind of not be amenable to anything chemical intervention. That's how they keep mutating. They do all kinds of weird things, and you're very, very hard to target them. And this protein was such that it didn't have any function at all it does was interact with different host immune system and try to prevent that signal from the host saying that that cell is infected, so the virus can keep propagating. It doesn't have any enzymatic function. It doesn't have anything. So it was very, very, it is very hard to target that protein, but we still took it up. We did a lot of assays on it, and we worked with, we have got good colleagues in IIIM Jammu, Dr. Ram and Dr. Parvinder, and we kind of worked with them and finally identified uh, three chemical leads, I must say, at this point of time, because it was able to interrupt in both, in general in vitro systems up to cell-based systems. We could find them intersecting the viral proteins. But yeah, we couldn't take, we are still working. But again, at that point, it became more of strategic because why would anybody want to invest in a HIV drug when there's something out there for 10 rupees? So
Mhm.
to take all those pros and cons as well. So we did that and we are still working on it. And we were supported by the government of India to do this work. And it was very nice to, embark back into drug discovery. And around this time is when I met Professor Ashok Venkatraman, who leads the CCBT. And
Mhm.
he was like, it's an in silo operation. Why don't you join the cell-based team here? And that's how I got endocytosed into the CCBT or absorbed into the CCBT. So,
Yes. We see a great journey, like a ladder, literally a ladder from this to this to this. So
yeah.
now, like in the last section of this conversation, let's talk
Mm.
about the things which kept you going for decades in research. Two things. First, all the challenges which tried to think you that you should give up, or you might have thoughts at some point of time, or the second part which always made you overcome all those thoughts, kick those thoughts out, so that you can enjoy your research as you are doing right now.
Oh yeah, I told you I started with the protein, which is now the greatest of protein, and it was really like doing a PhD again because you're dealing with all the stars of research stars, and they are the actual celebrities in India, and you had to tell them that hey, I have done my bit also. You have to do. It was kind of doing a PhD all over again. We were having monthly meetings. I used to go and talk with everybody, practically everybody in the team, and it was a completely new experience. And when every failed experiment or every small hurdle, we had to say why we couldn't do it at that point of time, and restart and go again. But the industry discipline did help a lot because
Mhm.
industry are used to working, we used to give like a project specific risk, and we
Mhm.
used to give what is known as a work breakdown. I didn't know that you could split a simple experiment in small bits and actually have it, that's what I learned in the industry, like have those small tiny bits done by different people, by myself or with my team, and you
Mhm.
could get the work achieved, and it had a specific timeline. And this work breakdown, you have to predict ahead of the project, not during the project. So
Yeah.
it was so that, and also project specific risk, like to what is the biggest risk in the project, we had to mention it, and how are we going to overcome it, and what is the cost of overcoming it, because it's industry, it's a startup. So you had to, really define to the rupee. You had to tell why you're doing those experiments and why does it make even sense. And in the industry, again, I had like very good mentors from who worked in GE and all. And this is what they taught me actually. And we also learned a lot of statistical packages, like it's called design of experiments. What are the key experiments that you have to do, to make this whole thing project a success? So
Mhm.
just do those one or two experiments. We didn't have to do 100 experiments. It will all be by doing things like design of experiment. You have to do those key experiments. So these kind of routines, I never thought because again, I told you it was a cross-disciplinary team. People were engineers, they were working across space, and they never understood why a cell couldn't behave the same way every day. So I was like, cell is a
Yes.
system. It has to work the same way. Why do you want to why do you keep saying you're a cell biologist? It has to work the same way. And it's not enough if Anandi is able to do it in the lab. It's a diagnostic kit. Somebody sitting in the middle of Madhya Pradesh, and the heat also has to work with that kit, and it has to work the same way for him. So how are you going to, dummify it, or how are you going to simplify it so that, anybody in the field can work the same way you can? Yeah, those were all huge, huge learning experiences. And so here I was coming back to academy. I thought I had it all, but nearly, really, the HIV proteins humbled me. So I was like, I can get this assay done in 6 months. I am good. I have done it all. No. Anything. And now I'm back to oncology. So none of these proteins, I mean, people are studying them for decades for a reason, trying to find the new drugs for them for a reason. It's not easy.
Absolutely. And I
Yeah.
think this is the thing when, like, most of this guidance is very crucial for the people who are in early career stage, so that they
Mm-hmm.
should know, as you mentioned, which project is going to give you definite results, which are more of the exploratory parts. So how they want to start their next stage as a researcher when they are transitioning from a postdoc to the faculty, or as you mentioned, even after decades of research either in academia, in industry, things keeps you on your toes. And that is the beauty of research, which keeps you, like, always fascinated. And sometimes it helps you humbling down also. You're like, okay,
Yeah.
I thought I was overconfident in committing about the timelines and whatnot, but it tells you that, okay, there is way more than what you think. And that is the
Yep.
beauty of research. Absolutely. So
Yeah.
now the
I
next,
can work.
yeah, yes, please.
So now we are actually working as a part of an international team. We are funded by the Gates Foundation, and it's actually for a completely different area again. It's for contraceptive, non-hormonal contraceptives for women. And it's again very amazing to be back in the international network. And as you rightly said, they have very strict. It could be a drug early target validation or early drug discovery, but the go no goes that you said just now are very very critical. And it doesn't matter if you say a no go. That doesn't mean the project is a failure. It is just
Yeah.
mean that target is not amenable for chemical intervention. That's all. So yeah.
And
So
absolutely, I think understanding this, what is a success, how do you define it, success of a research project? Because now with this fast-paced research globally,
yeah,
you sometimes it happens that you might have the best solution, but it is the pricing which you are looking at. If somebody can afford or offer it in half of it, then you might have the best technology, but is it scalable? And that's
yeah,
when some of the patents never get into the field, translational things. It is like, it is a great thing, great project. All the solutions were awesome, but because of the different limits. So in
correct.
your experience, have you experienced anything like that, that the research answer was there, but it is the technology which keeps it mellowed?
All the time. All the time. So in the industry we had a project, we had like something, we had like I told you, right, we had to come up with diagnostic solutions for instrument in making. And that lot of times so there was this thing called anti-nuclear antibodies. So it's for diagnosis of lupus and autoimmune diseases. People are so used to working with microscopes, and we had come up with a flow cytometry based solution, and it was supposed to be coupled with the instrument, but at the last time it was working. We got the flow based solution, as I told you, where we had our timelines. I was working with fantastic team, and we got it all the way, got a solution, got it working. We had a dried down cell based reagent that could work in the field, and it was time we could freeze cells and keep cells in room temperature. We came up with technologies to do everything. All of it is fine, but business wise it didn't make sense. We had to just drop the project and say, okay, I did my part, and that's it. And as I told you, always humbling. I thought I had it all, came back here, worked with NEF, and after a point we said, okay, fine, let's take a, because even if we inhibit one function of NEF, we are not going to inhibit the other activities of NEF. So let's take a pause here and come up with a different strategy. So yeah, it keeps evolving all the time, and that's what keeps you thinking, right? And that's how you build on your research and come up with the next solution. So
That's right. It's like always going process. It's always ongoing. Sometimes
yeah.
when, so for the last bit of it, if I can know your thoughts or experiences about it, because at the later stage of research career, the researchers have developed this thing that they cannot pause their brains even when they are sleeping. Their experiments are going on in their head, and there are all the stories. So, what were your experiences and what you can suggest to the middle career researchers, like who are transitioning, either just got their tenure or what? How should they take a break?
Oh, I can't tell you enough. People will tell me, even today I send in mails at 3 o'clock in the morning. And if people haven't seen me do that, I mean, you can ask any of my teams from across every section of my life. They've seen me do all kinds of crazy things. So, what I feel is you should have a hobby aside research, something else, something to think. For me, it's sometimes even my children. Sometimes it's your, some activity, some hobby, something that it could be yoga, something. I don't know what you do, but I do all of these things. For me, my children is a huge, a blessing. I come home, I sit with them, and it's sometimes very fresh to see their perspective, their thoughts. And sometimes they ask, even they know my project actually so well, they ask these questions, and when you explain to them, you get your ideas also. I mean, it's not that it has to be a break. Your back of your head will be going in these problems. But when you hear it or talk about it, or, go just do something else, you might get better ideas, is what I feel. And I really like the US system that way. They work hard. The Monday through Friday will be really hard work. They won't take a break, but the weekend, they'll manage to take that weekend off. And sometimes I think it is essential, even to progress. People think it's a waste of time, you shouldn't have breaks and all. I think it's absolutely necessary to get the next best idea. So
So
that's what
true. And I think,
driving. And, if you don't take that break, you'll be, at some point, you'll get burnt out.
Mhm.
You will not see that passion, and you should take that break. You should have something else to do also. So,
Absolutely. And I think we do talk about burnouts in our these conversations, that it is a real thing. And it is not like just because you are in research, in any profession you can be, burnouts are the real thing. And taking these breaks are so essential, as Dr. Anandi said, that the rejuvenation is important. Whenever you are rejuvenated again, whatever makes you happy. Some people like going to movies, others are more nature lovers, they want to go to a forest to get that energy back. And then once you have the fresh mind cleared out, because in cluttered brain, even the same experiment you might have done in 100,000 times, you might fail in even that if the brain is cluttered. So, for every single researcher, what is the most important part is clean and clear brain and the clarity of your research experiments. Obviously, troubleshooting is an important part, but as mentioned about those, if you have your experiments absolutely planned and then you have your controls intact, everything is working well. That's what ensures that what you are doing. Yeah.
Oh, one more thing, we write down. So the industry is also good that way. So we write down every teeny tiny bit. In fact, so people think that is the good, that's what is good lab practice actually, documenting everything. So that's key. If you don't document, then tomorrow you don't know what you have done, and you go back and uh, to even troubleshoot what you just said now is so important. If you don't write
No,
down things,
absolutely. I think without keeping a good journal, like writing everything down to the every single step, like some different people have different ways. Some people have their protocols printed out and then they just keep on ticking that, okay, this check, this check, this check. Some people actually write it every single time they do it, like manually. Everyone, because if you do not have a written thing, troubleshooting might be easy for tomorrow, but after a week, it is almost impossible
correct.
for any researcher to recall all those steps. So, having everything written down is very important. And this implies for researcher of any stage. You
Yep,
might be an undergrad researcher, a PhD student, a senior scientist who actually happen to be on the wet lab bench and working all of those. It is true for all of those. And the early you get the habit, the more you ensures more success in that. With this,
correct.
we'll say thank you so much, Dr. Anandi. And at Empowering Science Foundation, we believe in breaking the barrier. One of the barrier is the students feel this discontact. They're like, oh, Dr. Anandi is such a big, how can we approach them? So we always say to our audience that they can write directly to our researcher celebrities, or if they want Empowering Science Foundation to put in touch with them, we are there to help them. And we should create this niche where any person who's going through a rough phase can contact a celebrity. And when we say here researcher celebrity, it's like let's celebrate research. Research is there. If that makes you happy, we are here to help you to keep that happiness and share that with the world.
Yep, absolutely. Always welcome. And yeah, we have hosted more than 100 people at CCBT actually,
Mhm.
interns and early kids, career stage. So yeah, happy to come and have a chat anytime. Yeah.
Thank you so much, Dr. Anandi. And with this, we'll say to all the audience, stay tuned for another episode of Researcher Celebrity.
Thank you so much for this opportunity. It was a pleasure being